All right, so this is the slide we finished with, looking at the endomembrane system and key players in that system. My goal for you is to be able to visualize, generally speaking, the roles of the endoplasmic reticulum and the Golgi body in this process. How molecules are moving from one to the other potentially, and in the process of this movement of molecules, there are vesicles formed. So keeping in mind these three in red, conceptually to understand. So your book has a diagram similar to this. So here would be a nucleus where transcription would occur to make a messenger RNA. Out through a nuclear pore, this messenger RNA be on its way to, in this case, the rough endoplasmic reticulum. And so the rough endoplasmic reticulum has ribosomes. So the ribosomes will be used to convert that message, translated into the formation of protein. So translation will be happening on these ribosomes to form proteins. So these green squiggly lines are the proteins that were made. And sometimes the proteins themselves get used in the endoplasmic reticulum. There's a few descriptions of what they might be used for. Sometimes they stick around to help detoxify. But oftentimes the proteins are packaged into vesicles for final repackaging and refolding, maybe getting edited or modified at the Golgi bodies. So again, a vesicle from the endoplasmic reticulum transported to the Golgi body for final packaging and then sent to its final destination. Maybe it's to the cell membrane, maybe it's to be excreted from the cell. It just depends on what the protein is. Because this is a challenging concept, I do have a slide on the notes that has a visual of this process. It's not allowing me to videotape it because I think there's copyright issues, but it will be available on your slides. I encourage you to watch the 3-minute video because it really does describe this process in more of an action-oriented way as opposed to just the flat concept on a textbook page. So give it a look over. Another concept your book brings up is endosymbiosis, or what is an endosymbiont? Well, the two that are mentioned in your book are mitochondria and chloroplasts. So when scientists started looking at mitochondria and chloroplasts, two things became curious about the fact that these two organelles are about the size of your typical prokaryotic cell, and both have their own DNA. So mitochondrial DNA is a thing, so is chloroplast DNA. In other words, they have a plasmid ring of DNA very similar to that of bacterial cells. So scientists puzzled over why these cell organelles have so many similarities to bacterial cells. And so the idea is, or the proposed idea is, in the course of the formation of the first eukaryotic cell in time, that perhaps a larger cell engulfed a smaller bacterial cell, and they began helping each other out. And so these The idea is that it started out as a mutualism relationship between two different cells. Maybe one of those cells was better at extracting energy from food. What's now the mitochondria. Maybe some were good at capturing light energy and producing carbohydrate from that. And that became this relationship. So the idea is that today's eukaryotic cells evolved based on this idea that these two cells merged somewhere in time and formed a symbiotic relationship, and ultimately led to the specialization that we see in multicellular organisms. And in fact, they share several genes that are also in bacterial cells. Cytoskeleton is another section. The dynamic cytoskeleton, I think they call it in your book, page 68. There's some basic concepts for you to remember here, but there will also potentially be some extra credit questions for our eventual exam. So if you take detailed notes of this section, maybe you'll remember some of those extra credit questions when they show up at the end of the quarter. But ultimately, cytoskeleton is about giving three-dimensional structure and framework to your cells. They allow things to move within the cell, like motor proteins move vesicles through the cell along the cytoskeleton filaments. So again, just jot down these ideas, keywords here. Maybe take a few more notes on some of the extra stuff and learn a little bit extra. So we talk a lot about what cells are made of and what's in cells, but so much of what happens in our bodies is actually based on things that leave cells, that are exited from cells. So what structures form outside of cells? So when we talk about a cell wall, that's formed on the exterior of the cell membrane. It's the building blocks of that cell wall are excreted from the cell through its cell membrane and then scaffolded together on the outside. And then plants' cells also secrete a waxy cuticle, helping to protect the exposed surfaces of that plant, help it reduce water loss. Our cells, what we call these secretions that might come from any given cell, is called an extracellular matrix, or in plural, extracellular matrices. So for example, bone is the skeletal extracellular matrix that is excreted from bone cells. So most of your bone isn't actually a cell or a group of cells, but the extracellular matrix from bone cells. And a lot of what holds cells together in your body is because of the extracellular matrix that is created from those cells. So again, this is another section you can dig a little more deeply into if you want to potentially get some correct extra credit questions. Last slide here, just revisiting life's emergent properties again. What characterizes a living thing? They all have in common. So they make and use organic molecules. The 5 big ones that we talked about: proteins, lipids, carbohydrates, nucleic acids, and ATP. They, of course, have to consist of one or more cells. They are able to metabolize and undergo the process of maintaining an constant internal environment, as well as they can, the process of homeostasis. During their lifetime, they grow and develop, and they use DNA as their hereditary material. And they also have the capacity to pass on that genetic material from generation to generation, but also modify those genes by mutation, and eventually that may allow to adaptation within that population as environmental pressures change. So that concludes chapter 4 notes, and just again revisiting, we're talking about cells, and cells is the basic unit. Cell is the basic unit of life. We understand those emergent properties of life, so just revisit those.